Welcome to Plenary 4. Very interesting lineup this morning. Anesthesia, Psychedelics, and Consciousness. We'll wait, I think, 2 more minutes while people trickle in and start 8:35. Hope everybody's enjoying themselves and had a good breakfast and sleep. All right, then. It's my pleasure to introduce the first speaker from Wellesley College, is it, or university? I'm not sure. Wellesley College. Michael Wiest. He has training in neuroscience, physics, and his title of his talk will be Old Theory, New Evidence, Microtubules Are the Biological Substrate for Quantum Consciousness. Let's give him a round of applause.
Thank you. Good morning, everyone. I'm Mike Wiest from Wellesley College in the United States. This picture is meant to represent our hard problem, formerly known as the mind-body problem. We've got the physical world represented by the bricks, and the consciousness that somehow mysteriously pops out of that represented by the I. I'm going to talk about some experimental evidence that may be unfamiliar to you, but I also, by the end of my talk, want to tell you that we already do have a solution to this hard problem. So here's how I've organized my talk. The first part is going to be mainly about experimental evidence, and then the second part will be regarding some theoretical issues. And so I'm going to start by talking about anesthesia, and my modest result here about what a apparent target of volatile anesthetics in rats, and then some other sources of evidence regarding this quantum microtubule hypothesis. So starting with anesthesia. This is a quote from Claude Bernard, one of the early investigators or discoverers of anesthetics, and he noted that everything that's alive is sensitive to these same chemicals. So this chart from a somewhat recent review of anesthetic mechanisms is showing how, or it's listing candidate molecular targets of these anesthetics, but a remarkable fact is that the same gases, vapors that we use during surgery in humans work not only on other animals, but on invertebrates and even plants and single-celled organisms who, have no neurons. So in us and in animals, we talk about hypnosis. That's a code word for unconsciousness. Down here, we don't know if they're conscious or unconscious or what, but the effect of these gases is to immobilize them, stop them wiggling around, or slow them down. So on this table are a bunch of candidate molecular targets of these volatile anesthetics. People tend to focus on the ion channels because those are what govern the electrical activity of our neurons, and we know that in order to behave, we have to talk to our muscles via action potentials in our motor neurons. But there are a bunch of other candidates because these molecules bind promiscuously to many proteins and targets in biology. I'm not going to talk about all of them. I'm going to focus on the cytoskeletal elements known as microtubules. But because they bind to so many different things, the tendency in the field is to assume that the way they make us unconscious is by acting on some combination of multiple ion channels and other targets like maybe mitochondria and also microtubules. I'm going to point out there are some, in spite of that sort of current consensus in the field, there are some striking empirical facts that suggest that maybe instead these molecules primarily act on one main target. But before I do that, I want to quickly outline the two competing classes of models that we're trying to compare. So models of consciousness and models of how we understand how anesthesia works. So the classical picture is that it's a network of neurons firing action potentials, and we've got the quantum picture. In both of these models emphasize functional connectivity among different areas as opposed to just raw firing rates or activity levels. So everyone agrees that when you disconnect different parts of the cortex, you go unconscious. But the underlying reality is what the disagreement is about. So here are some of the remarkable facts I mentioned earlier that suggest that maybe anesthetics are acting primarily, or volatile anesthetics in particular, are acting primarily on one main molecular target. On the left here is the Meyer-Overton correlation or relation, which shows this pretty this relationship between the solubility of these different chemicals or molecules or even atoms in multiple cases. The solubility in olive oil predicts their effectiveness or predicts the effective dose that you need to put someone unconscious. And the reason that this suggests a conserved target, a common target, is that these, and moreover, it suggests that it's more of a physical interaction rather than a chemical lock and key kind of typical drug binding interaction. What suggests that is that these molecules have little or nothing in common chemically. As I said, some of them are just elements. And so it's hard to understand what chemical lock and key kind of property they could have in common. And so that suggests rather that it might be some physical interaction. On this side is a plot showing that you can combine different anesthetics. If you give half of an effective dose of one and half of an effective dose of another, you tend to get approximately one effective dose. And the examples in this plot are chosen because the pairs are chosen to have different effects on particular ion channels. So for example, one of these might strongly modulate the activity of GABA inhibitory receptors in the brain, and this one might have no effect on those receptors, but nevertheless, you put half and half together and you get a full dose. That suggests that that receptor can't explain the total action of those molecules. And the study that came from, moreover, concluded that even no combination that they could envision could account for this additivity of effective doses of different anesthetics. So what could that main target be? You know what I'm going to say. This is a report with several of our most valuable players on the author list. So what they showed in this theoretical study was that the binding of several of those molecules correlates, the binding and the extent to which those molecules disrupt resonances in the tubulin that makes up microtubules correlated with the potency of those molecules as anesthetics, and also predicted some molecules that were not anesthetics showed that they would not be according to this model. So what I'm saying is this study more or less reproduced that Meyer-Overton correlation by assuming that the main target is in tubulin. So everybody here, I think, has heard about microtubules. these tiny protein tubes inside all cells, and I've already mentioned they're made out of tubulin subunits. They're in all cells, but in neurons they have very specialized organization and accessory proteins called microtubule-associated proteins that hold them together with these cross bridges, but can also be phosphorylated and change their functions. So they are part of the machinery that governs whatever microtubules do. I've got one slide, because I'm trying to be brief, to stand for all the amazing things that microtubules do that show that they are not just tent poles, they're not just railroad tracks or bones for the cell. They have many amazing functions, and this is a familiar one, the beautiful dance of the chromosomes where they're being pulled apart. It's the microtubules that are pulling them apart, and it's a microtubule organizing center made out of microtubules that seems to be choreographing that dance. And as Dr. Hammeroff has mentioned many times, single-celled organisms use microtubules and other parts of the cytoskeleton to crawl around. And there's also fascinating evidence that they can use their microtubules to detect even electromagnetic signals and even the direction that they are coming from. But I don't have time to go over those studies. But this was just to say that we already know microtubules are more than tent poles. Okay, so coming back to anesthesia, here is my modest experimental contribution where we used this drug called ipothyolone B. It is an anti-cancer drug. Most anti-cancer drugs are microtubule binding drugs because cancer is runaway mitosis, and you just saw that the microtubules are involved in that. So they use microtubule binding drugs as anti-cancer drugs, but most of them don't penetrate the blood-brain barrier. But people have started to develop ones that do penetrate the blood-brain barrier to fight brain cancers. And we got one called EPO-B and gave it to our rats and simply showed that it took them longer to fall unconscious under isoflurane anesthesia than the control animals. So the way we did that is measured the latency, the time it took them to fall asleep, so to speak, as loss of righting reflex. That means when you flip it, when it's on its back, or you flip it on its back, it doesn't immediately turn back over. If they're awake, they definitely don't want to be on their back, at least not from you doing it to them, maybe from their buddy. And so we're simply timing how long it takes them to lose their righting reflex under isoflurane with or without EPO-B. And here's what the data look like. Again, average lower latency is loss of righting reflex latency, and there are the EPO-B animals with the significantly higher. It was 69 seconds long. It took them 69 seconds longer to fall unconscious. So we can think about how we could account for this finding in terms of either of these two competing models. I'm going to start with the classical model. So it is conceivable under the classical model that messing with microtubules could put you unconscious because I hadn't mentioned yet that but Dr. Hammeroff has already mentioned that they act as railroad tracks for synaptic vesicles and other materials that need to be sent to different places in the neuron. And so one of the critical things that they carry around are synaptic vesicles to bring out to the synapses, to release, to communicate with other neurons. So you could imagine if you mess with the microtubules somewhat, maybe that would disrupt this transport of synaptic vesicles to the synapse, and reducing synaptic activity could plausibly reduce neural activity, which would lead to functional disconnectivity that either model understands as leading to unconscious. And as it happens, there are a couple studies showing that isoflurane does affect synaptic transmission, but those studies investigated and determined the mechanisms by which isoflurane was affecting synaptic transmission, and it had to do with mitochondria which are intimately related to microtubules and synaptic factors rather than microtubules. So what I'm saying is, although this drug affects synaptic transmission, it does not seem to be via this effect on microtubules. Before I go on to evidence about the quantum model, I wanted to mention this study by Emerson et al., where this is now not using the kind of volatile anesthetics I'm emphasizing or focusing on, but they designed their own chemical to target microtubules in particular, and showed that messing with microtubules is sufficient by itself to at least cause these tadpoles to stop swimming around. Don't know for sure if they're conscious or what. So now, coming back to my paper, I'm just skipping to the bottom of this abstract where we come now to the quantum idea, where this was the result, and I said this finding is predicted by models that posit consciousness as a property of quantum physical state of neural microtubules. And I thought it'd be worth pausing for a moment to here to say what is the significance of asserting that consciousness has to be quantum rather than classical? In my view, the primary motivation for that hypothesis is that the quantum model can solve a fundamental problem that the classical model cannot solve, and it is not a very difficult problem to understand, does not require math or technical jargon. It's the binding problem, expressed here from a neuroscience textbook, exactly how the parallel streams of sensory data are melded into perception images and ideas, remains the holy grail of neuroscience. It's kind of suggested by this portrait of his wife by Dali, where somehow the whole that we are arises out of individual pieces. How does that happen? So to be a little more concrete, I see that little table or stool or whatever it is. It has a certain shape that's represented by neurons in one part of my brain and a color that's represented by neurons in another part of my brain, but somehow I experience that as a whole. The question is, how can that be, and how could such a holistic thing actually guide behavior or affect behavior? So here's how William James, a famous American psychologist, expressed this binding problem. Take a sentence of a dozen words and take 12 men and tell to each one word. Then stand the men in a row or jam them in a bunch and let each think of his own word as intently as he will. Nowhere will there be a consciousness of the whole sentence. So that's not a sentence. So the suggestion is that if we have a macroscopic quantum state in there, that could solve this problem, because I'm going to go into more detail about this later, but the reason for this is our conscious states feel like complex wholes. There are no irreducible wholes in WHOLE. There are no irreducible wholes in classical physics. Quantum physics is all about irreducible wholes. That's what a quantum state is. So you've all heard this, but let me quickly review that quantum theory describes probabilities of different measurement outcomes with a so-called wave function. In between measurements, the system evolves deterministically and locally, and you can think of the Schrödinger equation for concreteness. At the moment of a measurement, the wave function changes discontinuously to represent not many possibilities, but the single one that got chosen. This is nowadays contentious, this first note, that the wave function is objective, not epistemic. I don't have time to argue about it right now, I think, but I'm taking this to be true. We see it's non-deterministic, and what I want to emphasize is the non-locality of it. Non-local coordination among distant parts of a system, that is irreducible holism. Here's just the illustration of the idea of the wave function collapse, also known as quantum state reduction. Technically, wave function only refers to when we're talking about position in space, but this idea applies whatever observable we're talking about. You have some range of possibilities, you make a measurement, and then you get one answer. But you're also aware that quantum theory, the textbooks don't tell us what a measurement is or what causes that collapse to a definite outcome. So the objective reduction point of view says the collapse really happens. It's not some artifact of learning new stuff. That's why we're calling it objective, as opposed to epistemic or subjective. Here is the famous undead cat of Schrödinger. I'm just bringing it up, although it often gets brought up, to remind you the fable is meant to show you, to tell you that quantum theory, if you leave out the measurements, you get absurd predictions like the half-dead cat, which we don't observe. So that's why we don't want to throw out the measurements, even though they're ugly and problematic. So here finally is ORC-OR, the specific quantum theory that I think is closest to being right, has the best chance of being right. ORC-OR, orchestrated objective reduction, associates each quantum collapse in non-living matter with moments of so-called proto-consciousness, tiny meaningless flickers of existence. But biological organisms have evolved microtubules and other structures to orchestrate those moments of collapse into larger scale wholes that carry information that's useful to us. So, I'm not going to dwell on this because Dr. Kurian has already talked about his own result in detail, but the upshot is that the reason this has been considered implausible for so long is that our technological attempts to create quantum effects in the lab so far have tended to require super cold temperatures, and we are not super cold, so people thought that was ridiculous. But now Dr. Kurian and colleagues have shown us that microtubules exhibit this quantum superradiant behavior even at room temperature, and moreover, it gets stronger as you join them into larger and larger structures, which is the opposite of the prejudice that when things get bigger, they get less quantum. This is one slide I took from Dr. Bandyopadhyay's, one of his papers, Ghosh et al. 2016, just illustrating some of his methods where he's recording membrane voltages and stimulating microtubule resonances at the same time. So these are not just correlations between microtubule resonances and membrane voltages. These are causal demonstrations that, first, that microtubule resonances control membrane voltage, which is necessary. If ORC-OR is going to be true, the microtubules must be able to talk to membrane voltage because, again, membrane voltage is how we move our muscles to behave. We have direct evidence that microtubules drive membrane voltage. But moreover, he's done this across multiple neurons, and that's another critical requirement for this theory to be viable, is the quantum entanglement or coherence must be able to span multiple neurons. I think many people are shy about saying that because it still sounds implausible to many people, but that must be the case if we're going to solve the binding problem. So I'm willing to say out loud that there must be a macroscopic quantum entangled state in our brain that spans centimeters, not just microns, because, again, different features that we experience as a whole are represented centimeters apart in the brain. So I think everyone has heard of that work, but it seems that not many people have heard of this work, where now we're talking about direct biophysical evidence of a macroscopic quantum entangled state in the living human brain. This is one of the papers that came from these two guys, one of the more recent ones, but they have a series of them. They set their regular old MRI machine to unorthodox settings designed to detect entanglement. They detected an MRI signal that looks like the EMG of a heartbeat, the so-called heartbeat evoked potential. But they argued there's no way that the MRI machine should be able to pick that signal up. And they made a case that it implied entanglement, that there's no classical mechanism for that signal. I've only found one person disbelieving what they said, but that person did not provide any classical mechanism as an alternative. So as it stands, I take this as evidence that there is a quantum state, a macroscopic quantum state in the brain, the living brain. And moreover, one of their subjects fell asleep in the scanner, and they saw a degradation of that signal. And they also did other experiments where they correlated the fidelity of that signal with working memory performance. And so based on that, they conclude that it's not just there, but it's related to consciousness and cognition. I have to be quick about this because I want to get to the second half of my talk, but I feel this is something that not many people are aware of as well, that there's a large literature about quantum-like behavior of people when we answer questions. And people have derived these identities or inequalities about relationships between answers to questions, statistical relationships between answers to questions when you ask them in different orders, and also all sorts of irrational conclusions that we come to seem to be described within a quantum formalism. People have started trying to figure out how you might make a classical brain do that. I'm asserting my opinion that it doesn't look that promising. Another thing that I don't have time to go into much detail about, and I'm sorry I don't have time to introduce this active inference theory, which is a dominant paradigm in cognitive neuroscience right now. Friston, the author of that, the inventor of that, recently published a paper where they developed a quantum version of this active inference formalism and derived that quantum entanglement and quantum computation must be widespread in biology. Nobody seems to have noticed that paper. Another thing is that within this active inference framework and also just in real life, we know that our moments of experience are discrete. They come in discrete, non-overlapping chunks. And again, I'm sorry I don't have time to go into much depth about that, but I think we can take that as a firm conclusion based on experiments like backward masking experiments, where a stimulus that comes after an initial stimulus can prevent the first one from being perceived at all. And there's a large literature about those kind of effects that imply that for every conscious moment, we integrate over some finite time to generate that conscious percept. And you can see that must be true, or we would not be able to perceive motion. So, ORC-OR, by associating moments of collapse with moments of consciousness, naturally accounts for those discrete cycles that we know exist. Whereas this theory doesn't have a way to naturally make them happen with classical neural mechanisms. I argued that in a paper that's under review at Computational and Structural Biotechnology Journal right now. Okay. So I want to turn to what you could call some theoretical problems or conceptual problems, because I'm not going to show any simulation results. So this kind of theoretical work you could call conceptual, or if you're a neuroscientist who wants to dismiss them out of hand, you could call them philosophical. And then I would use William James to defend myself. So, does the quantum model, does ORC-OR solve the hard problem? In other words, does it explain why any particular physical process should or must come with consciousness? There's the representation of that hard problem. Do you know the answer? The answer is no. The OR is a physical process. Dr. Penrose has said that. And it seems to me that no twists and turns of logic can get you from properties that are just about movement and space to qualia. So, no, just asserting the quantum model does not solve the hard problem. But if we allow ourselves to postulate a mental property of physical systems, then because OR solves the binding problem, it makes that a viable step. Okay, people refer to that as panpsychism. I know Dr. Hameroff doesn't care for that term. We can quibble about that perhaps later, but so by solving the binding problem, I'm saying the quantum model makes panpsychism viable as a solution to the hard problem, and finally brings consciousness within the scope of science. I want to reassert or re-argue that that's not an option for the classical model. So the classical model could also just postulate a mental property of matter or of physical systems. It's not cheating to do that. We have regularly introduced new fundamental properties into our fundamental theory. Charge, then new quantum charges, spin, color charges. We do it all the time. This would be the first time we introduce a non-physical property. That's okay. But if you try that with the classical model, it's not going to work because, again, even if you give the classical system consciousness, which I'm representing by the color here, it is still all reducible to neighbor interactions, so that you can eliminate whatever the holistic thing is, the conscious experience that you're trying to explain. You can completely eliminate it from your description of this system without losing any descriptive or predictive power. This is Chris Fields, who spoke yesterday, made a similar argument that if you try to go panpsychist in a classical model, you can only get, or he was focusing on a functionalist model in particular, you can only get mind dust. That's a term from James again. This is the bits of mind dust. You only get the little pieces because there are no larger scale irreducible objects in classical physics. I know you may doubt that, so I put it all by itself on one slide. If you doubt it, just reflect that every classical physical law is a differential equation. It's written in terms of dx's and dt's. The dx's are infinitesimally separated neighboring points in space. And so if you imagine that, or if you postulate that a certain pattern of activity in the brain comes with this consciousness, it can't do anything because you can eliminate it from the description by reverting to the local description, and you've got a complete description of the behavior of that thing. So it must be epiphenomenal if you take that route. This is the inconvenient truth. I think if we want to be able to explain how our consciousness evolved so that fire hurts and a ripe apple tastes good, the classical models are ruled out because the consciousness can't do anything, can't provide an evolutionary advantage. I know you're thinking that it's the same problem for the quantum theory. But first I want to mention aside from the huge advantage of the quantum model in accounting, or in solving the binding problem, accounting for the unity of consciousness, there's also potential practical advantages or evolutionary advantages of having unified quantum states to work with. There's a tendency to think that our memory capacity, if you Google around for what's the memory capacity of the human brain, you come across estimates where they assume, if a synapse can be in two different states and you have 100 synapses, then you have 2 to the 100 memory states. But that's not true within a classical framework, because the Hopfield-like networks that we actually use to model memory are linear, have linear capacity in the number of neurons. So if you have 100 neurons, you might only be able to store 15 or so patterns, 1/5. And that increases only linearly as you increase the number of neurons. And that could be a real issue, because it is demonstrated that we have amazing memory and it lasts 100 years. Well, there's this tendency to think that quantum computation is hyped because there's only a couple useful algorithms and they're only useful for robbing banks. So that pertains to speed, perhaps. But speed is not the only possible advantage that a quantum system could have. Since 2000, and now this field has developed further, so other people have proposed similar models. These guys married a quantum search algorithm called Grover's algorithm with a neural network architecture and came up with a quantum associative memory that has up to exponentially growing capacity with the number of neurons, as opposed to the merely linear capacity of the classical models. So there's a quantum memory advantage potentially if you've got a quantum brain. So, but does the quantum model succumb to the same epiphenomenalism problem that I described for the classical model? Everybody thinks so, because you can imagine the quantum model without the consciousness. You can conceive of the zombie, as Chalmers talks about it. But I want to distinguish two aspects of this epiphenomenalism problem. One is that conceivability of zombies. I'm going to say that's not a real problem, because I can conceive of a horse that uses dragonfly wings to fly, but I also know it's impossible. So you can conceive of things that are impossible. That's not really a problem. The scientific problem is, can we account for the evolution of this phenomenon? So the problem, it seems to be, consciousness can't alter the physical dynamics, so we can't understand how it could confer an advantage and evolve. The solution is to recognize a necessary relation between the unity of consciousness and the objective quantum unity of the physical substrate. Then we can understand why conscious creatures have an advantage. We can predict which systems will be conscious. We can relate the structure of their qualia to the structure of the entanglement that is the basis of the unity. So I'm suggesting that the psychophysical bridging principle that Chalmers was seeking in his 1995 book is unity. And I'm representing this this is an actual bridge called Unity Bridge between Mozambique and Tanzania. And I like the horns or the tusks, because I think in mythology that represents the transition from the material to the spiritual world. But that's my suggestion, is that a necessary relation between a property of the qualia, or the consciousness experience, and the physical substrate is a way to account for how our consciousness could have evolved. And I go into more detail about this in my recent paper in Neuroscience of Consciousness. So here's what I said. Microtubules do a lot more than hold up the tent. I described ORC-OR. I argued that by solving the fundamental binding problem, ORC-OR makes panpsychism viable as a solution to the hard problem. So that's why I say we do have a solution right now. And the solution to the evolutionary phenomenalism problem is to recognize a necessary relationship between the unity of experience and the objective unity, the physically efficacious unity of the substrate. Thank you.
How did you get invited to this conference anyway? Just curious. Let's take a few questions. That was an excellent talk. Thank you very much, Mike. Let's have a few questions. Anybody? This gentleman here, who's got the, oh. All right, yeah, let's start here. And we'll move backward.
Thank you very much. It was very interesting. I think you expand this model to another level.
Thank you.
My question is, do you think the consciousness is not just in the brain, it's beyond the brain, and this effect is practically is like an antenna who enable us to reach other level of consciousness with our brain? Not for human, also for other animals, etc. Thanks.
Yeah. I don't think it's ruled out, but when people make the analogy of the transistor radio and not being able to find the music is coming from the music station. And I can't imagine what the station would be that would be sending these thoughts. And moreover, it seems that we find correlates of anything we look for any experience we look for the neural correlative of in our head, we generally find it there in the neurons.
But just yesterday, there was a lecture about memory is not just in the brain, it's in a quantum space. So in this case, we can connect to the quantum space for the memory, as example.
It's conceivable. I don't see the evidence for it so far.
Over there, and then you.
Could you elaborate a little bit on the evidence that this supports for panpsychism?
Evidence for panpsychism, I guess the only evidence is that I'm conscious. I'm a physical system and I'm conscious. And it seems incoherent to me to say that making something that has only physical properties, which again, physical property means a property that can affect motion, no matter how complicated you make that, it seems clear to me that you can't transubstantiate it into experience. So the fact that I'm a physical system with consciousness suggests to me that physical systems have consciousness.
All physical systems.
That all physical systems have a conscious property or a mental property, but it requires biological orchestration to make it into something interesting like we have.
Over here.
I'm just interested, this notion that anesthesia targets the microtubules. Is it possible that that is just suppressing the expression of consciousness? Because if, when you're unconscious, you're still dreaming, there's still some conscious activity going on?
Absolutely. So when you're dreaming, I would not call you unconscious.
Probably there's no dreaming under anesthesia. It's controversial because of propofol coming in and out. But and one previous question I would call it quantum pan-proto-psychism.
Yeah, a little more technical, you could call it pan-proto-psychism, where it's not asserting that everything is conscious, but everything has the potential to be conscious if you put it in the right arrangement.
Two more questions. Back there, yeah.
So really appreciate your talk. One question related to your comment just now saying that it requires biological systems. So we had this really interesting conversation last night at one of the panels where we went all the way from kind of human self-aware and so on down to microbial into kind of more cosmic, planetary or whatever, right? So what I'm starting to really appreciate here, the first time I heard about microtubules, I was like, well, but I'm a microbiologist, so bacteria have a very significant awareness and they don't have microtubules, but they have tubulin-like structures, which are also quite complex, and they have aromatic rings and so on. But my starting to appreciate the thesis of this org-OR is that certain degree of complexity of quantum uncertainty gives rise to an ability to capture a certain complexity of maybe the consciousness concepts. So it's not necessarily biological, but we've evolved to have this very, very complex ability to have very concentrated this quantum, let's say, uncertainty space within a very tiny physical material in there that we have such a concentrated conscious experience, whereas a chair, which also has a few kilos of material, doesn't have that degree of complexity of, let's say, capturing. That's kind of where I'm evolving with my understanding of the science being coming out so intensely, but I wonder what your thought on that. I just reflected on your need of biological.
Thank you for asking that, because I did not mean to imply that it must be biological. Just so far, that's the only way it has happened, but it's a physical effect, and so in principle, you should be able to make it happen in other ways.
Please be brief.
Thank you, Mike. Excellent presentation. I'm really kind of wedded to the whole concept of how this works, and it's very exciting. But at the same time, we still know that there are a few things to be cracked, like the hard problem, where qualia fits in, and how that is experienced. Where would you see this orca-or having to fit within maybe expanding the box of its metaphysics in order to really understand how all the aspects of conscious experience can be explicated?
I'm not sure if I'm understanding your question, because you started by saying we haven't solved the hard problem, but I think if we postulate proto-panpsychism or pan-protopsychism, that is the solution. I know it sounds too easy, but it's not easy, because we still have to figure out the details of how physical systems relate to.
Let's have one more quick question here, then we have to move on. This gentleman here in the second row, please, and then please be brief, because we're a little bit behind.
I'm just curious if you could speak to any evidence that the epiphenomenalism problem of consciousness is a problem, in the sense that what does consciousness do? Do we have any evidence to suggest that it does anything for the purposes of evolution or human behavior? I mean, there's a lot of studies. Ben Libet, obviously, with the kind of famous studies of conscious perception coming way later in the game. A lot of the work that Robert Sapolsky from Stanford has put out shows that we don't really do anything with consciousness. It's just something that sort of happens. So is epiphenomenalism an actual problem that we need to solve for?
The reason I think thank you for asking that. The reason I think it's a problem is it really appears that our consciousness evolved because, as I said, fire hurts. It would be a massive coincidence. That's very good advice, to take your hand out of the fire. It would be a massive coincidence if it played no role in getting you to take your hand out of the fire, but it happens to give that good advice. And so maybe I meant to emphasize that the version of pan-protopsychism that I'm advocating here does not allow any interaction between the mental and the physical properties. I want to emphasize that because an interactionist theory might not be ruled out, because physics is not causally closed, but I suspect that it's actually incoherent to say that a qualia causes something to move, and so I wanted to show you that there is a solution to this problem, even if we don't resort to incoherent interactions.
Okay, let's thank Mike Wiest for a fantastic talk. Thank you, Mike. And it's my pleasure to introduce the next speaker, Pär Halje from Lund University, how consciousness may depend on brain cells acting collectively. Evidence from rats. Pär.
Okay, good. Okay. Thanks for inviting me to this very interesting conference. I've had a lot of inspirational talks today and yesterday. I'm going to talk about how consciousness may rely on brain cells acting collectively. And this evidence comes from my research on rats. So I give LSD to rats. That's my job. I thought that might require some explanation of like how somebody ends up doing that. So I think I was in my early 20s or something when I took my first acid trip. And I was studying theoretical physics at the time. I was very interested in the nature of reality and the nature of consciousness and so on. And it was really quite inspirational for me, this experience. I kind of changed my subject from physics to neuroscience. I really wanted to understand this amazing organ and how it's constructed. But it was also a bit discouraging because it became obvious to me that studying consciousness scientifically is really not possible, right? I mean, it's not within the reach of the scientific toolbox, basically, to study consciousness. So I thought also, yes, I will study the brain, but consciousness is probably best studied on a personal level by experiencing and reflecting on your own consciousness. So I ended up doing more practical studies on the brain, like studying how different sensory modalities are integrated into a common representation, how motor programs are learned and selected, and so on. I've done a lot of Parkinson's research. And actually, it was this Parkinson research that kind of drew me back into the scientific study of consciousness again. So I will start my talk about that, actually. So we have models of Parkinson's disease. Parkinson's disease is caused by dopamine neurons in your brain dying. So you get too little dopamine, especially in a region called the striatum here. So this region is supposed to be black when you color it properly. This side is healthy, this side has Parkinson's. So these rats have Parkinson's on one side of the brain. We just wanted to study what the brain is, what the electrical fields in the brain look like when you have Parkinson's. So we implant these electrodes, these microwire electrodes in the brain, and record. And we did a very, like an unexpected finding that when you have a Parkinsonian rat and you give them this drug levodopa, which is converted into dopamine in the brain, they develop a side effect of the medication called dyskinesia. So it's not, well, Parkinson's, as you might know, is lack of motion, right? You move slowly, you have a hard time initiating your movements. Dyskinesia is the opposite. You get involuntary movements. So it looks a little bit like spasms in a sense, but it's not. It's actually involuntary movements that happen, and it almost looks like you're doing some weird kind of dance. The rats do this also, but what we found was when these involuntary movements happen, you have this really weird phenomenon, a narrow band oscillation in the electrical field of the brain at 80 hertz exactly. And this oscillation should not be there. This is not something that a normal brain displays. So we were really fascinated by this. And a few years later, people managed to implant an electrode strip onto the motor cortex in humans with Parkinson's and record, and they actually found exactly the same oscillation there, only present when they have dyskinesia. So it was like one of these translational markers for dyskinesia. About at the same time, another group of researchers were looking at ketamine and found a similar oscillation. You can see it here in the rat nucleus accumbens. So the nucleus accumbens is a part of the brain which is, it's the ventral striatum, also related to the part involved in Parkinson's. So they found it on ketamine, they found it on MK801, which is a related drug, and later also they found it on LSD. It's a bit weaker at a bit lower frequency, but it's definitely there. And this really got me interested. So because this is just too weird of a coincidence, I think. You have this similar phenomenon. It's a narrow band oscillation in the cortex basal ganglia system. Different frequencies, different neural modulators, sure. But the thing is that the cortex basal ganglia system is organized like this. So you have the cortex feeding into the striatum and deeper basal ganglia structures, feeding into thalamus and then back to cortex. So it's this kind of circuit. And the theory is basically that this circuit can become hypersynchronous in some pathological states. And that's what we see here. So maybe then it's the same phenomenon in both these cases, that you have like a modulation of the serotonin system in this case, or the glutamate system, and you also get these hypersynchronous states, but in a different domain of this cortex basal ganglia system. It's not the motor domain, it's the more cognitive domain. So then I was like, oh, maybe the psychedelic state is like dyskinesia, but it's like cognitive dyskinesia. You get all these thoughts and ideas coming into your mind without you actually willing them actively. So this gave me kind of a handle into studying consciousness, I thought. So if we have this type of clear phenomenon in the brain that we can study, then we can actually give psychedelics to animals, go in with all the sharp tools that we have for animal neuroscience. We can't implant electrodes like this in humans, of course. But still, they have the same receptors. They seem to react in the same way to these drugs in some way. Because otherwise, we have some known neurocorrelates of consciousness in the literature. The most famous one is probably the brain waves, discovered originally by Hans Berger over 100 years ago. He invented the EEG machine, basically. So basically, you can see on these brain oscillations with electrodes just on top of the skull if you're sleeping or awake. In animals, you can go in a little bit deeper and pinpoint what's happening. So you can, for example, stimulate a region in the brainstem called the reticular activation system and kind of force the system out of the sleep state into the awake state. And you can also do the opposite. You can stimulate in the thalamus in a certain way to kind of trigger this sleep type of activity. So it's really tightly linked to conscious awareness, right? The other one already came up in the previous talk, the feature binding problem. It's often presented like this, that you have different aspects of sensory information that needs to be combined dynamically. There's no time to actually create new synapses and so on, so you have to have a more flexible way of combining this information. And one way of doing it is to fire the neurons in sync or out of sync to a common carrier frequency, which you can find in the visual cortex, for example, the gamma oscillation around 40 hertz. So somehow then this information, when fired in phase with this oscillation, it can be integrated, and the theory goes then that, well, this integrated information becomes part of conscious awareness. Well, a little bit of primer on psychedelics, what it is, the pharmacology and so on. We have classic psychedelics, LSD, psilocybin, DMT, mescaline and so on. They can look quite different. They have different skeletons, so to speak, the tryptamine, phenethylamine, ergoline framework. But they all bind to the same receptor, basically, the serotonin type 2A receptor, 5-HT2A. And we know this since the 90s, experiments in humans where you could selectively block this receptor, and you can also block the effect of the psychedelics. So it's this receptor that is important, even though like these molecules bind to a lot of receptors. This is a key mechanism. But we also have other types of psychedelics, dissociative anesthetics like ketamine, PCP. They don't go on the serotonin receptor directly. They go to a glutamate type receptor called the NMDA receptor. We also have other types that goes on other system, but I won't go into that now. But basically, we don't know based on the molecule or the receptor binding profile if a specific molecule is a psychedelic molecule. We have to ask people for that. We have to, So there's questionnaires developed for this that that ask about people's experiences. Say, okay, if you have a high score on this, it's a psychedelic. Because the thing is that there's also, for example, 5-HT2A agonists that are not psychedelic. So yeah, there's no way to know. And just as an example of the type of questions that you can ask people, I think this is kind of funny. So here are some questions you can ask. I felt especially assertive. I experienced a dissolution of myself or ego. I felt more important or special than others. Some of these questions here are supposed to target ego dissolution, and some are supposed to target ego inflation, like I felt especially sure of myself. Okay, ego inflation. And if you plot the answer on these questions to the dose of LSD, for example, you get like a linear relationship between ego dissolution, but not so much for the ego inflation. But if you take other drugs, you can get actually the opposite curve. So I mean, it's funny, but it's also kind of fascinating. I think that we have molecules that does this to us. You know, modifies our personalities, and in a sense, yeah. But what is special with psychedelics, I think, is that it changes the fabric of conscious experience, not only changing the content. So we set out to replicate these studies on psychedelics and rats and try to expand it a little bit. And the goal basically was to find a specific neural activity pattern, as specific as we could, related to the psychedelic experience. Well, we don't know what the rats are experiencing, but you know what I mean. And importantly, this activity pattern should be similar for all types of psychedelics, independent of the site of action, right? Because it has to work for all the different kinds of psychedelics, not just one kind. And the activity pattern should not be present for the other types of psychoactive substances that we have. For example, the stimulants like amphetamine and cocaine and so on. And also importantly, the activity patterns should not trivially just depend on behavior. So for example, if the animal is just running around more when we give one type of drug, well, maybe we're just seeing the neural correlates of running, not the correlates of a psychedelic experience. The tools that we use are these microwire electrodes. So here we have like a Frankenstein rat. The thin hair-like structures are tungsten wires that go into the brain, and we have screws to anchor it in the skull. And we have to build these electrodes by hand under the microscope, and when we're done implanting it, we can do a CT scan and locate where the wires actually ended up. The signals we are recording are, they look like this basically. So here's an electrode in tissue, and the other side is connected to the ground. We get this kind of wiggly trace with some spikes on it, and we can separate this signal into two components. The low frequency part, it's between 0 and 300 hertz. This is believed to be related to the group activity of many synapses. So when a synapse is active, you're opening ion channels in the receiving end, the postsynaptic end, and these currents are creating this, if it's like enough of them, usually like 10,000 or so. These sharp, quick things, we can get to those by high-pass filtering, thresholding, and then sorting these spikes. They look like this if you zoom in. And these are action potentials. They're about 1 millisecond long. And they come from the active opening and closing of ion channels across the membrane. And this is like the pulses that neurons use to send information to each other, right? Yeah. So the study design is very easy now. Once we have this electrode implanted, we just give a bunch of different drugs after recording a baseline, and then we see what happens, and then we repeat the experiment a couple of times. So it's completely. Let's see if this plays. Yeah, it's completely spontaneous behavior, spontaneous electrical activity. And this rat now is actually on LSD. I don't know if you can tell. It's not the nice colors. Sometimes no, they actually listen to minimal techno. We play but seriously, we play a series of click sounds to it sometimes to kind of probe the auditory system. It's terrible music. But the only thing you can see that tells you that this rat is on LSD is that it sometimes does this little shake. It's called a head twitch response. It's like, it's doing this. It's a natural behavior, but it's much more frequent on serotonergic psychedelics. So that's the only sign that we actually know that the drug is doing what it should. So yeah, we're doing, like, apart from the electrophysiology, we're tracking behavior with video. We have an accelerometer on the head to pick up these shakes, and we also score behavior manually. And we did find some behaviors, of course, that were specific to the different classes of drugs. So this head twitch response is specific to the serotonergic drugs. We have like ataxic behavior, so they basically have a hard time controlling their movements. They look a bit drunk, especially on the ketamine type. And then they are, of course, very hyperactive on the stimulants. They just can't stop. But we don't really find a common behavior for both classes of psychedelics. But that's, in a sense, a good thing, right? Because then we know that whatever common patterns we see with these two types of drugs are not trivially related to a behavior. So I start with the spiking data, the firing rate modulations. So here you basically see each line here is one neuron. So we have about 300 something neurons here in total. This is the activity on the baseline, and then the activity after the drug. And red means more spikes, and blue means less spikes. So you can see that there's like a variety of responses, both excitation and inhibition. But for the serotonergic drugs, we have actually a dominance of weak inhibition. It's like light blue mostly. And for both the ketamine type and the amphetamine, we have a dominance of excitation, actually. And just by looking at these pictures, you can see that actually these two look more similar than these, right? And we can quantify this, of course. We can figure out, based on the waveform, with what type of neuron it is, if it's a principal cell or an interneuron. And we, of course, know where the wires are, so we can split it into different parts of the brain. And we see that this inhibition, due to the serotonergic drugs, are widespread. It's in all regions, all cell types. But for the NMDA type of drugs, we have actually excitation of the interneurons mostly. So we can quantify now the similarity of this firing rate pattern between the drugs with something called the Bhattacharya distance. And what we see here then is that the amphetamine type and the ketamine type are much more similar than the LSD type and the ketamine type. So this is not really a good neural correlate of the psychedelic experience. But we did find these oscillations, right? And it turns out that, yes, they are specific to both types of psychedelics, and they're not really present in the amphetamine case. And they're really strong. I mean, you can really see them with your naked eye here. But this is a spectrogram now, so you have frequency on the y-axis and time on the x-axis. Yeah, it's really strongly enhanced. So we could also, of course, look in different parts of the brain and see where does this oscillation occur. And again, the pattern is similar between these two types of drugs. It's mainly in the prefrontal parts of the brain, the medial prefrontal cortex, orbital frontal cortex, ventral, yeah, but also in these basal ganglia structures, the cognitive parts of the basal ganglia, the ventral striatum, and the olfactory cortex. So, what is the relationship now between firing rate and these oscillations? Where do these oscillations come from? I mean, it must be neural activity that are creating them, right? It turns out that it's not single neurons that are firing at this frequency of about 150 hertz. Each neuron maybe fires once every 10 cycles or 8 cycles or 17 cycles or whatever. And you can see this in the autocorrelogram of the spiking. So this is one neuron now we're looking at, the probability of a certain delay between spikes, right? So if this neuron would be firing regularly at 150 hertz, you would see like a spiking fence-like shape on this one, but it's quite flat. So the oscillation somehow seems to appear on a population level, not on the individual level, on the level of the individual neuron. But we can see in individual neurons that each cell has a preference for firing at a certain phase of this oscillation. So here you have the LFP, the raw LFP, the filtered LFP, and the dashed lines here are when a particular neuron is spiking. So if you pool all this data together, you get this histogram. You say, okay, what is the preferred phase on baseline and during the drug? And you can see this kind of wavy pattern here, which indicates that the neuron has a preferred phase. So I think this is an important point, that at some we can only understand this now as a system-level property. It's not an individual neuron. The system-level property might still arise from properties of the individual neuron, but the phenomenon can only be observed at the system level. But the most surprising thing, I think, that we found was the phase relationship of these oscillations in different structures. So you can see here now these LFP traces in different parts of the prefrontal cortex and the ventral striatum, and they do look kind of similar in some structures just by looking at the amplitude modulation, and you can quantify that here. That yes, we have within structure it's quite big, but it's also significant between structures. The frequency, I don't know if you noticed that in the spectrogram pictures, the frequency is not exactly the same, it's wobbling a little bit as well, but it's always wobbling together across structures. So again, it seems to be a global phenomenon. But if you zoom in on one of these and really try to look at the phase relationship between them, it's a very tight relationship. So here's like a spherical, like a histogram of the phase relationship between this black electrode trace and the other ones. And you can see that it's a quite tight relationship and sometimes a little bit shifted, but it's shifted, a few degrees basically. And this corresponds to a delay of maybe a millisecond or so, a few milliseconds. And the weird thing with this is that to go from this structure to this structure, you have to pass at least 2-3 synapses. And the time it would take to do that, it's at least 10 milliseconds. You have to send this action potential along the axon, you have to convert the action potential into synaptic activity, again convert that into a new action potential and so on. That takes time, about 10 milliseconds. And 10 milliseconds is that's more than one cycle of this 150 hertz. You know, one cycle is 6.5 milliseconds or something. So it can't be just one group of cells projecting this signal to everything else. It has to be something more sophisticated. And I believe that actually this system has become a system of coupled oscillators. And a coupled oscillator system, it's studied frequently in physics. It's basically a system of individual oscillators, represented here by these metronomes, but they are connected. They can interact weakly. And as you see here, they stand on a table, and this table is actually hanging from the ceiling, so it can wiggle. So a displacement of these weights of one of these will actually displace the table in the other direction, right? So they can influence each other weakly. And if you do this right, you will see that they, after a while, assume a common phase. Because, yeah, the common phase is the most stable state, in a sense. Yeah, so we basically think that different parts of the brain has this tendency to innately oscillate at a certain frequency, and under certain conditions you can couple them together to do these phase synchronous behavior. You have a few rebels still here, but they will soon give up. Don't worry. Okay, anyway. But we also have to show, of course, that we do have individual oscillators, and we have some evidence of this. And the way you can see this is that, I mean, yeah, before I showed you that, like all these traces were in phase, but you also have examples where they are in anti-phase, and that's a sign that you have a little current dipole generated locally in the tissue between the two electrodes where you measure, and then you get this phase inversion. Yeah. So I will actually conclude there and say that, yeah, the most specific neural activity pattern that we could find was these high-frequency oscillations at around 150 hertz in the prefrontal cortices and the ventral parts of the striatum, and the olfactory cortex also. And of course, I mean, I think it's reasonable to assume that this type of hypersynchrony has severe effects on cognition. We can only speculate exactly how, but I mean, yeah, we know that neurons use phase synchrony to communicate, to open and close these communication channels, basically. And I also have a hard time imagining how you know, causality in such a system. You know, if you think about, like, you have information in a certain part that is supposed to be sent to another part. How is that working when everything is just in this coupled oscillator state? Also, of course, I mean, do we really need to consider quantum coherence when we actually have electric field coherence? Isn't this solving the same problem? We have a field that is spread across the brain. Why can't we just use that to integrate information between different parts? Another interesting potential implication here is that this hypersynchronicity is probably causing synaptic plasticity in the system. So it's a classic way of inducing synaptic plasticity, actually, is to stimulate two connected neurons at a high frequency. What fires together wires together, right? It's one of these Hebbian learning rules. And we know that synaptic plasticity is involved in the therapeutic effect of psychedelics, for example, in depression. So this could be part of that mechanism, actually, that you induce synaptic plasticity that later can be used for changing your patterns of thought or patterns of behavior. But of course, we still don't know if this phenomenon exists in humans or if it's just an effect of something peculiar in the rat nervous system. And the reason why we don't know is that it's very difficult to measure this high frequency in humans. You basically have to be inside. You can't do it with surface EEG. Before I end, I would like to just make a little show you the resources we have. So you can go check this data in the publications, of course, but actually all this raw data is available freely online if you want to do your own analysis of this, and maybe you have an idea of how this works and you can actually do this yourself. And soon we will also publish an atlas for LFP oscillations, like a neurophysiological atlas that we can use to explore these type of signals even more. And these are my collaborators. Thank you.
Okay, we have time for a few questions. The gentleman right here, let's get the mic to him, please. Please be brief in your questions.
I'm thinking of the surprising findings of Imperial College on using, you mentioned humans there, but the psilocybin studies on humans, where it surprised the research team there, Robert and Carhart Harris, where there was an attenuation in the default mode network, but similar to your hypersynchronous, there was crosstalk in the brain in a synchronous way that wasn't expected far beyond, and there's a beautiful colored rendering of that. And your research seems to be in parallel with that. And I'm just wondering if you've had chats with them on that.
Yeah, and I actually even borrowed some data from this. So they did an MEG study with psychedelics, and I tried to look for this oscillation there. But it's I couldn't find it. But it's a bit dual actually in our own data as well. We do find increased synchrony in some frequency bands and decreased synchrony in other frequency bands. Because it's also a common finding in the fMRI studies that you get like a decoupled network. So I think it's both. It's depending on in what band you're looking.
Over there.
In laser physics, there's also synchronization of modes, and where the slow modes are the dominant, slaving the faster modes. Did you see also a dependency on the frequency? Your oscillator were all at the same frequency, your model. Do you work also with different oscillators?
I mean, in the local field potential is kind of limited upwards, right? Like if you go up, you don't really have the same type of signal anymore. But it is this oscillation seems to be actually modulated by lower frequencies. Around 5, 6, 7 hertz or so. Okay. So there is definitely a coupling there.
Yeah. Any other questions? Yeah. Where? Okay, two more, him and then him. Okay.
Thank you. That was very interesting. I have a small question, not so important to your main message, but I'm curious about the head twitch behavior that you said was also a natural behavior. Do we know if it's associated with anxiety or what it might be associated with in the rats?
No, we don't know. We don't know. No, it's but it's like a brainstem motor program. That's all we know.
Thank you.
Yeah.
Right behind, second row there. Last question.
Yeah, thanks for the talk. My colleagues at the Qualia Research Institute have been working on a model of coupled oscillators to describe brain dynamics, but it was motivated by phenomenology, just with psychedelics, for example. And I'm curious, because you mentioned having some experience with psychedelics yourself, whether you've experienced some of these coupled oscillator dynamics yourself and maybe some of those experiences.
Yeah, I don't know what that would be like to experience that, but yeah, I don't know. Maybe I'm experiencing that right now. I don't know. It's a difficult question, but yeah, I mean it's a very common experience, of course, when you ask people like that you feel connected to everything basically on these peak experiences. Yeah.
Okay, let's thank Professor Halje for a great talk. Our next speaker is Professor Penberthy from University of Virginia, and I'm going to find the title of her talk here while she's coming up. Psilocybin and prolonged grief disorder, role of subjective experience on outcomes. Professor Penberthy, thank you very much.
Thank you. I'm very happy to be here. Can you hear me okay?
No.
Now?
Now.
Wonderful. And thank you for your patience. I know this has been a long time. Please feel free to take a bathroom break if you need it. I had to right before this. So thank you. I will be talking about psilocybin in humans. And so we're shifting gears here a little bit because I am a clinical psychologist, not a neuroscientist. And a lot of the research I do is based on my clinical practice and my interest in healing people and communities and societies. So I was trained in the clinician practitioner researcher model where I research what I apply and I teach that, and I research and teach and apply it clinically, and it's an ongoing sort of system that helps me fine-tune what I'm doing and specialize to help people. My early interest, interestingly, were about a lot of these altered states of consciousness. Not so much exploring it through medications or medicines, but more through meditation. I was taught early on transcendental meditation and had early exposure to people like Raymond Moody, and took this interest in contemplative practices and altered states of consciousness and did some more formal training. So I've accomplished the psychedelic assisted psychotherapy training, become a board-certified clinical psychologist, and I'm very interested in taking these concepts and applying them in the world. I've also had the good fortune to be at the University of Virginia for my entire career at the Division of Perceptual Studies. For those of you who are not aware of this division, it was founded in 1967 by Ian Stevenson, who was the chairman at the time of the psychiatry department at the University of Virginia. He studied children who remembered past lives and developed this institution really to study the idea that human consciousness could survive the death of the physical body. And even to this day, these are some of the areas we study. So a lot of what we look at are ways to understand and measure consciousness outside of the physical body. So looking at things like near-death experiences, after-death communications, out-of-body experiences. And one of my colleagues who studies that is here at the conference as well. So we really are trying to find out whether mind and brain are distinct and separable, or whether consciousness survives physical death, and to explore the impact of this on humans and society. As a clinician, I'm particularly interested in helping people who have some pathology. A lot of the work I do is in the Department of Psychiatry and Neurobehavioral Sciences, as well as the Cancer Center. And as you can imagine, working with cancer patients and their families, I see a lot of death, dying, and grief. And one of my areas of research is looking at prolonged grief disorder. Prolonged grief disorder, for those of you who don't know, has recently been placed into the DSM, the Diagnostic and Statistic Manual of the Psychiatry Association. And it is definitely a growing problem, unfortunately, in our world. As you may surmise from wherever you come from, we are rife with war, disease, COVID didn't help, and natural disasters. So the rate of prolonged grief disorders is increasing. It's characterized by something quite different than depression: yearning for the deceased individual, identity dysfunction and disruption, functional impairment, and importantly, the symptoms in the DSM have to last 12 months or longer. Unfortunately, we currently have no gold standard treatment for this disorder, not only because it's only recently in the DSM, but because it's been traditionally mistaken as PTSD or depression, and it's really quite different. It really focuses more on this maladaptive longing and preoccupation with the deceased, coupled by that difficulty integrating the loss. There's a hypothesis in some of the prolonged grief literature that some of this may be arising because of humanity's sort of distancing from, at least Western cultures, distancing from any kind of spiritual tradition or religions. And thus we approach the death of a loved one with no context, no sort of framework to understand it. And we know that from our work looking at prolonged grief and the brain, the neural correlates of prolonged grief include some disruptions in the default mode network. As we saw earlier, this is sort of an area where some of the psychedelics do target. And for refresher, we've got areas here that the default mode network nodes include, which we heard a little bit about. That was quite convenient to have that right before us. So we did a little research looking at understanding exactly what psychedelic treatment might work for prolonged grief, and came up with the psilocybin because of the mapping of that on the brain activity in the grieving brain. What's interesting about prolonged grief, and you can even say many other psychiatric disorders, is that the diagnostic criteria lumps everybody in together, and we may have many different people with prolonged grief, but the individual aspects of those symptoms depend on that particular person and their experience, their history. So what we see in people with prolonged grief, they meet criteria for it, but the aspects of the grief may be different. They may have unanswered questions that can no longer be answered because that individual's gone. They might be longing for connection, feeling alone in the world, abandoned. They might have concern for that individual who's deceased, for their well-being, or wondering where they are, feelings of remorse, and now they can never make it up to that individual, or larger existential questions. This is a problem because we do see fairly significant rates of this in individuals, 7 to 10%, and that number's increasing, of bereaved adults. So someone who's lost someone will experience these sort of symptoms. And among children and young adults or adolescents, we also see this, and it's increasing. And this, to me, is heartbreaking. Prolonged grief in a young person can impact the trajectory of their life, and in a very negative way. And as I said, it's scary because rates are increasing, and it doesn't look like they're going to go lower anytime soon. So the study that we designed is a first for our university. So we have to be very careful. We have to go very slow. The University of Virginia, for those of you who do not know, is a quite conservative university, a state school in Virginia, Thomas Jefferson's University. And we are the first to do any kind of psychedelic research. So we decided to be very conservative. We are doing a one-arm study looking at 1 dose of psilocybin for individuals who meet criteria for prolonged grief disorder. We are also very interested in looking at their brains. So we're doing a functional MRI before they take the medicine. They obviously enroll in the study, and then we do the fMRI. We are doing a grief elicitation that is developed at Texas Tech by the Singer Lab. Very elegant grief elicitation, so we can see the brain, the grieving brain, when the grief is elicited and when the brain is exposed to an image. It's specifically an image of the loved one who is passed on, and then also we're looking at the brain when exposed to images of another family member and a stranger. And we're getting that before the psilocybin and after the psilocybin. Importantly, we are also collecting not only quantitative data, things like we've heard about, so their experiences on mystical experiences scales and other measures of grief, but we're also going to be looking at the microphenomenology of their experiences. So we want to see what first of all, macrophenomenology, what do they experience? And then microphenomenology, how does that consciously unfold in a moment's time? And we will be looking at follow-up up to 6 months. So we chose psilocybin again because, as you've heard, the disruption of the rigid thought patterns. The rigid thought patterns are quite predominant in prolonged grief, and psilocybin has demonstrated that ability to break those patterns and might help eliminate some of the ruminative behaviors we see in these patients. Increasing neuroplasticity. There's evidence that this will promote some neural connectivity, as we heard, and synaptic growth, so helping hopefully reorganize some of the grief-related components we have in their neural. And increased openness. So we know that psilocybin pretty reliably increases that trait of openness, the personality trait of openness that can be associated with cognitive flexibility, creativity, and receptivity to new perspectives, which obviously would be helpful in addressing the grief and integrating that. We know from studies that come out of places like Johns Hopkins, who we've collaborated with, that the transformative experience, the mystical experience, or what we're calling a subjective experience, is very important. And we want to really dig deep into this idea of looking at what are these experiences that people have. There's some evidence that a mystical experience is required for the psilocybin to be effective. But we're going to look a bit differently at it and look at individuals' specific experiences. So our hypotheses are basically that the psilocybin, the 1 dose, integrated in the way we have the study designed, will help with symptom reduction acutely and will promote some psychological change that we're measuring, specifically things like openness. And we are incorporating this in a classic way where we're introducing the medication or the treatment with an intention. So in psychedelic-assisted psychotherapy, you work with the patient to set an intention for the treatment. Ours will obviously be related to the grief, but it will be specific to the individual. And this is very important. We are not, however, doing psychotherapy with the individuals. They are required to have a support person themselves, a therapist, a counselor, a chaplain, somebody who's already working with them, and they will return to that individual to help unpack some of this. We, however, will work with them to set the intention, administer the medicine, and work through the integration in a few sessions after the medication is provided, where we will really look at the macrophenomenology of what happened and the microphenomenology. But I'm very clear that we are not doing therapy. We are not doing psychedelic-assisted psychotherapy. We are really exploring the subjective experiences that these individuals have. And we still speculate that this will be of help. So we are going to be looking at those experiences. I'm calling them subjective experiences. You could think of them as mystical experiences. We will be looking at those as predictors of response. We're also, as I said, going to be looking at the neural correlates and exploring those. Now, it's going to be a small N. We only are looking at 20 people, and we've certainly not reached that yet. So we are limited in what we can analyze and do. It is interesting, though, and we're quite proud of this idea, that we are doing more than just the traditional randomized controlled trial where we identify a patient population, measure their symptoms, give them a drug, like a black box, and say, okay, some magic happened, and now we measure them again. And so the magic worked or it didn't work. We're really looking at what is happening to the individual. So we're doing that with integrated methodology, really looking at both the brain imaging and also these different levels of phenomenology. Looking at mechanism exploration. Yes, is mystical experience important? And what kind and how and to whom? And then really this focus on prolonged grief that's been neglected for so long. We are also targeting a young adult population because of the importance of catching this early and helping to save lives that way. The outputs obviously will be the clinical data that we hope to see shift in our measures of grief and the impact of grief. We will also be looking at things like psychological openness, flexibility, and other measures, and the neuroimaging findings. Again, hopefully being able to look at the pre and post in these individuals. And the qualitative insights, to me, are the most interesting. We really want to see what these individuals experience, how it relates to their set intention prior to the experience, and how that relates to the impact of their symptoms and outcomes. Looking specifically at that subjective mystical experience and how it helps them expand their consciousness in ways that are therapeutic. What experiences might impact change, what actually changes. And our hypothesis, as you might have inferred, is that there will be a mechanism that is similar to all, sort of like an umbrella, called this altered state of consciousness due to the psilocybin. And yet the content of what actually happens in those subjective experiences may be very unique and different for each individual, and we hope related to their intention and thus related to a positive impact on their grief. We know from our research, we did years of research prior to the study, just developing theories on which psychedelic we want to use, which population would be most appropriate, and looking at the different subjective experiences that individuals have on psilocybin. And this is just a list of a few. There are probably as many as there are people. We know that there's ego dissolution. We've heard that already. out-of-body experiences are quite common and interpreted in many different ways by individuals. A sense of increased connectedness, both social connectedness, connectedness as in I'm dissolving into the universe. There's often intense emotion, either a sense of emotional release, a sense of intense love, awe. There's a scale that measures awe by David Yaden. We're using that as well. Many people report contact with ancestors or ancestral lineage, or even the deceased loved ones specifically. And of course, there are encounters with other entities, deities, other magnificent beings. So people may experience one or all of these, or none of these, or other things we've not heard. So we are going to be capturing that. And believe it or not, in most current research and past research in psychedelics, this is not typically collected in a systematic way. So we want to really collect this information and hear each individual story and collect it in a way where we can look at the impact of the outcomes. We believe in many ways that this can become what is quite trendy in medicine nowadays, a tailored treatment, which means the treatment may look the same at the outset, the same dose of psilocybin, the same sort of layout of how this protocol is followed, and yet each individual's treatment or reaction is unique. So, for instance, in some of our work, we've seen that specific subjective experiences during the psilocybin are related to the people by their stories. So you may have someone who has a sense of yearning, feeling disconnected, and the impact of their experience, their subjective experience, may be a connection with the deceased. So maybe they had their psilocybin experience and they had a visit from this person, and they now feel a connection that they had thought was lost. And this is what they needed. You may have someone who wonders, I've heard this so many times, where are they? Are they just gone? Is it like they never existed? This is very hard for humans to feel, that someone is just absent and gone and not here anymore. And so that reassurance from a contact, from a sense of connection, whatever that might be specifically for the individual, may be very profound in what they need. If there is a sense of just depression, of just sadness, of this dysthymia, then perhaps someone's mystical experience of contact of love, infused love just sort of surrounding me. I hear this too. I just felt full of love all around me. This may be what that individual needs that helps address the issue that they've got. And of course, you might have people that say, you know, my loved one's dead, it's over, there's nothing, this sort of nihilism. And they may have an encounter with a creator that was very impactful for their sort of symptomatology. So we will be looking at these in the macro sense of what each individual experiences phenomenologically, and looking at the microphenomenological components as well. There is a methodology called microphenomenological interviewing. Has anyone here heard of it? Raise your hand if you have. Okay, we've got a very, I'd say 2%. Well, this is becoming more popular in this kind of work, in consciousness work especially, and therapeutic work, because it's a way for us to try, in a qualitative way, examine what is happening, how consciousness is unfolding. So we will be taking these particular macrophenomenological experiences that are happening. So let's say I feel awe. We can measure that quantitatively. We will do that. But that is that's sort of a sterile measure. We will then, in a microphenomenological way, work with them to understand and describe that experience for them, that unfolding in just sort of fractions of seconds of what was happening, what they were experiencing. And there are ways that we can do the qualitative analysis to explore that. So our idea is really to help understand what specifically is happening at that level as consciousness unfolds and changes. And again, going back to the fMRIs, look at what may or may not change there. And do those sort of look similar, which might be a hypothesis that perhaps changes overall look similar across these individuals, and yet their experiences may be quite different and would not have been captured in an imaging study alone. So as I said, the potential clinical impact is a novel treatment for a disease or disorder that currently has no standard gold treatment, and understanding the mechanism, which is a rare thing in clinical psychology and psychiatry. We rarely understand the mechanism. So this is a long shot, but we're going to give it a try. Enhancing our clinical practice. If you have prolonged grief right now, you're sort of out of luck. There's really not much that folks can do and not many people trained to help you. And we hope this will restore hope and give people another chance. As I alluded to, part of the reason why this is so important is because we have ongoing issues that create death, and specifically situations that are more likely to create prolonged grief. Prolonged grief happens more in people where they have a loss that was unexpected or violent. And we are certainly in an era where we see a lot of that. Unfortunately, for every death, there is an estimate that we leave roughly 9 times as many people bereaved. So we really do have a significant problem on our hands if all these people are left to bereave on their own without any treatment. Because the other thing we know is if you have prolonged grief disorder, again, remember, this is grief that's gone on for 12 months or longer that has functional impairment. It does not get better on its own. So these people will be potentially impaired the rest of their lives if it's not treated. And to me, that's just unacceptable as a clinician and a researcher. So we hope to return in the future with more of our data. The collection has been significantly delayed due to issues in the US at this point. But we do have funding, and we do have our medicines, so we will be moving forward. And I want to say thank you for your time and attention. Thank you to my amazing research team that helps me do this. They're listed here. And thank you for your attention.
We'll have a few questions for Dr. Penberthy, and I think we have some time for the other 2 speakers if you want to come up here after a few questions for her. This gentleman there.
Well, thank you, Dr. Penberthy. Always appreciative, eternally so, of the work of the Division of Perceptual Studies. And for those not aware of what they do, the work at the University of Virginia, take a peek. It's of the human condition in its most basic.
And there's that website right there. Yeah.
There you go. But I'm particularly appreciative of the emphasis that your study is placing on the subjective. With the psychedelic renaissance, there are those with the profit motive of venture capitalism that are trying to design the molecule to design out the mystical experience. And I'm hoping that your study will show that that's folly of pure physicalism, that you will show that it'll be a benchmark of that. So very much looking forward to the results and appreciative again of the work done there and the rest of the team.
Thank you.
Thank you. I have a comment and a quick question, a comment for you and actually one of the speakers, which is I would invite you not to use the term dissolution of the self. Even though Carter Harris has popularized it and so on, I can give you two examples of things that can be considered dissolution of the self that are diametrically opposite. One person with depersonalization starts experiencing, I am no longer solid. I don't feel myself, and it's a terrible experience. Number two, you have a more mystical-like experience where you feel I'm becoming part of it. You can call both of them dissolution of the self. So I think it is a very imprecise phrase, and I would invite you to consider, you know, with the rats you can't ask them, even in Lund, I'm from Lund, but to consider using other terms. And a specific question, historical, Freud in his Mourning and Melancholia paper hypothesized that people who would end up developing melancholia, which would be prolonged grief disorder, developed it because they had an ambivalent relationship to the person who had died. Has there been any research, as far as you know, that would either ratify or rectify his long ago hypothesis?
Thank you. That is a very helpful suggestion, and I think you're correct. We need to be precise in our terminology. I'd love to talk to you about what your recommendations would be, because I think you're right that that can mean different things to different people, and we need to be clear about that, so thank you. And with respect to the relationship with the individual, a lot of that work that I'm familiar with myself that I've done is looking more at relationships with individuals after death and exploring whether they were close or not close or had a difficult relationship or a healthy relationship. And we don't really see, to my data so far, any significant impact of those. The grief, prolonged grief seems to occur more in individuals with a loss that was sudden, whether or not it was someone that they were having an argument with or close with. It's sort of this unexpected nature of it, or the sort of traumatic component. So I'll give you some examples. The majority of the people enrolled in our study so far are individuals who walked into the suicide of a child. And so there is a significant trauma component as well as grief. And so, you know, you walk into the suicide of your teenager, of course you have a very dynamic relationship with a teenager, most likely. These, you know, anyone who has children knows that. And we think it's more that suddenness, that trauma of that loss. So, you know, it makes me think of what just happened in Texas. Just horrible, horrible flooding that wiped out a children's camp, a girls' camp. Most of them deceased now, dead from the flood. And I suspect many of their parents will be suffering prolonged grief if we don't have a treatment for them. So that's the sort of individual person we're looking to help. But that's a very, very interesting question. Thank you.
Yeah, thank you for this beautiful talk. I noticed that in the pictures you have chosen to show persons, many of them, most of them were women. Did you find differences? Are there differences in the number of women with respect to men who suffer from this disease, and the way they stay in it, and the way this potential therapy treatment works?
Yes, so the research data does indicate that this prolonged grief disorder happens more frequently in women. So I can't remember the rest of your question, I'm sorry. What was the rest of it?
If there is a difference in the way women and men stay in this disorder, if they develop different ways, besides the numbers.
Right, right. That's a very good question. That's sort of a broad question. So if I understand you, is there a different way they experience prolonged grief? And I think maybe we will see that. I don't know that right now. Like I said, because we typically say, here, fill out this questionnaire, check these boxes, and then you're lumped into, okay, you have prolonged grief. And we don't ask a lot of subjective questions. So I'm hopeful we will find that through the treatment. Do many of these women perhaps wish for connection? Or do they, and maybe the men suffer more remorse? I mean, I don't know, that's a hypothesis, but that's what I hope to see. Are there nuances that we see? My hunch is that it will be much more specific than that it will be very individualized based on that individual and their relationship with the deceased.
I suspect there would be very important differences.
Thank you.
Back there.
Thank you very much. I very much appreciate your encompassing approach, including a mixed methods approach. And I would be interested on your qualitative analysis. Did you code your data? And did you quantify the code frequencies perhaps? And did you triangulate qualitative data results with quantitative data results?
So this is all still in process. We are using the sort of a thematic analysis for the macrophenomenology and then the microphenomenological analysis that is for the microphenomenology. And because it is a small n, we are sort of still exploring how we will look at that along with the imaging data. So that's still in process, but we will be looking at that as well as the quantitative data. Yeah, I hope to bring more back where we will really get into some of the details.
So I think you might have said that, so sorry if I just didn't catch it, but are most of the people that you need to give treatment are people that already have a view, an understanding of, let's say, there's not something beyond the material world. So, they just see very materialistic philosophy, let's say. So what the drug you give them makes them see that there's perhaps another dimension, and that gives them some kind of hope, let's say. So the groups that you're treating, are these all people that have that view of the world, or do you have something, let's say, more mixed and they all need the treatment?
That's a really good question, and so I have two answers. We have a general assumption from the literature about the decline of belief in religion or spirituality. Many people nowadays in the West endorse either that they have no belief system like that, or that they are spiritual but not religious, and we really don't know what that means. Religious doctrine, following religious doctrines is decreasing. So we are going to, we're not really measuring that. I mean, we know sort of globally what that looks like, but we're not individually asking people, are you a physicalist? Are you, I'm not sure, and some folks would understand what that meant. But we will be looking at individual people and their experience of that grief and their intention, so that, for instance, we can see, do they have a sense that this person has just disappeared, turned into dirt, worm food, and And that's their concern, that they're nothing now, that we would be able to measure that and look at what changes with the psilocybin. Do they have a sense of continuation of that soul because they had contact with it, or they had contact with some sort of deity that now they have changed the way of thinking. So we'll be looking at that individually in a way to sort of measure it. So it's sort of getting at it in a backwards way without asking them specifically. Because the other thing I've noticed is in those questionnaires where you ask for religious beliefs or spiritual beliefs, many people do not know how to answer that. And it often leads to basically just information that you can't make any sense of. You know, I've seen questionnaires where people go through and check every religious belief, you know, like, and I'm not sure what to do with that. Or when they say no, I have no beliefs in religion or spirituality, I'm not sure what to make of that. So some of those I've honestly found to be a little bit challenging, and I'm hopeful that the individual level may have better predictability and help us understand. That's a very good question, though. Thank you.
One more question. Please be brief.
Are you talking to me or them?
Thank you for the talk. The question is, did you see any repeated subjective experience of the same person over time with the same trauma? Did they have a repeated subjective experience? And also, once they were healed, did these mystical experiences stop?
Once they
healed, once they were out of the grief.
Oh, okay, okay. Yeah, well, I'll answer that first. I mean, most of these research experiences, we don't measure that long term. So we only have knowledge up into the 6 months. And we will be looking at that. We don't know that yet. Will these continue? I think that's a very interesting question. We know if we teach people different meditative or contemplative practices, there usually is a drop-off unless they continue. But psilocybin works differently, and presumably there may be more of a permanent, or permanent being maybe years, change. So that will be very interesting to see over time. And the first question was... Okay, yeah. I mean, we are seeing, sort of thematic similarities. And so that's why we are sort of looking at groups of these thematic and macro phenomenologies. So we think maybe humans are programmed with a sort of a menu of options there, and we do keep seeing those pop up. Yeah. Thank you.
Okay, thank everybody. Let's thank all three speakers, Jennifer Penberthy, Mike Wiest, Pär Halje, and we'll be back 11:10 for the next session. Thank you.